if you guys go into research, this is going to be a huge topic. I don't know if you guys get the impression sometimes that all the science is done with certain issues. It is not done when it comes to the brain and the immune system. They are two really fascinatingly linked systems in the body. And integrating them together is a huge frontier right now. If you really think about it, immune cells are a lot like nerve cells. They have chemicals that they talk to each other with. They're different subgroups that have roles in this wider system, and it makes a lot of sense. They both respond to. They both utilize hormone structures. It's just a they're both very communicative cellular systems, and it turns out that we're learning that they continually interact together. And so let's talk about all the different immune cells that are popping up in the brain, or that we're finding out about. Obviously, they've been there all along.
If you open it on yours as well, we can just kind of both have it accessible. Okay, so basically what you're looking at right now is a really cartoonish cross-section of the 3 different types of cells that are involved in neuroimmune reactivity.
That we know of.
There's one more type of cell that's not on here, which is actually an innate immune cell, which is a mast cell. But of the 3 cells that are here, you have astrocytes, which are also known as astroglia. And these are cells that have these long pseudopods, and they wrap around the vasculature that comes into the brain. And so they basically are intimately associated with part of what is known as the blood-brain barrier, which is the interface between the vascular system and the parenchyma of the brain. Parenchyma is basically just the functional stuff. You can have the parenchyma of any kind of organ. You can have the parenchyma of the pancreas, you can have it of the liver, you can have it of the brain. That's where the stuff that does the work is located. Everything else is supporting infrastructure. So astrocytes...
You want to show the astrocytes maybe while we're at it?
I want to give this little piece, and then we'll go to all 3 of them. So there's astrocytes. There's the ones that hold onto the vasculature. There's oligodendrocytes, which are the ones that occupy the role of the Schwann cells in the peripheral nervous system. And so in the peripheral nervous system, you have these cells that wrap around the projections of your neurons. You have these myelin sheaths that protect the electrical propagation through the nerve. So Schwann cells are in the periphery. Inside the brain, these cells are called oligodendrocytes. And then microglia are kind of these mobile cells that move all around the brain. And these 3 cell types together form basically the neuroimmune system. In terms of microglia, you want to cover microglia?
Sure. I actually put them in a different order.
I see. Okay, you want to do astroglia first?
Yeah, those are the ones you mentioned first. But I wanted to say one thing for people who haven't studied neuroscience at all so far. If you think about the cables that connect your computer right now, they're not bare cables. This is really important. And so in order to ensure that they're not shorting out, that the electricity isn't just conducting to ground all the time, you have to insulate them. And that's a huge job to do in the brain, because all of these axons are essentially single cell wires, and they need to be insulated from the rest of the body at all times. And when they don't do that job, you get all sorts of problems, including almost everything we're going to talk about today, but also basic stuff, multiple sclerosis, right?
I mean, you have enormous dysfunction.
Yeah, you can have all sorts of neuromuscular problems. Which makes sense, because it's like if you nicked one of your wires on the ground, or we have all these wires everywhere in here. If you nicked one of them and it touched to something that was conductive, you would have a huge problem in your system. So it makes sense.
Right.
So a lot of these cells, cell types contribute to that process.
And for a long time,
and astroglia are a primary example.
Right. And so at the time when the brain was considered to be immune privileged, which is the fact that the brain was not being surveilled by the immune system, it was thought that these were largely structural and nutritional cell types, right? So astroglia come into contact with blood vessels, so it's obvious to be like, well, they're kind of the trafficking cells of the brain and they're bringing nutrients in, and then they pass them to the oligodendrocytes that are feeding the neurons, and then the microglia are
structural, maybe?
Structural. It was unclear.
Yeah, who knows? Yeah.
But now what we know is that the astrocytes are basically the regulators of immune function in the brain. They're involved in lots of things that stimulate the immune response in the brain, and they're also involved in a lot of things that downregulate the immune response in the brain. So having a healthy astrocyte response to the presence of antigen is really important, because you need them both to ramp things up when necessary, and you need them to do the opposite when things need to be calmed down. And so they are basically effector cells. And so these cells that are able to do high-level activation or high-level deactivation of responses are cells that are considered effector cells. And they produce cytokines. We have a slide a little bit later that talks about all of the things that they make. They can also be structural cells too, though, which is that if you have an injury in the brain, what will often happen is that astrocytes will migrate to the site, and they'll basically encapsulate the injury site in order to prevent it from leaking into the rest of the brain. And so depending on conditions, astrocytes will behave differently, and study of this is still very, very rocky. I'm not totally sure why, because people have been looking at the brain for a really long time and have been doing single-cell studies on the brain for a long time, but maybe a long time, 20 years, isn't really enough time. What do you figure?
I think they do have a huge structural role, and if you think about it, it makes sense. Like, we were talking about this too with respect to the primary and secondary lymphoid organs last time. It's hard to walk around and set up a bunch of cabling if you don't have some sort of scaffold to do that on. And if you look at these cells, they make a wonderful scaffold. And all of those little protrusions, those little arms that they have, those can have myelin at the end of them. So they can insulate, they can act basically like rafters in the brain to hold things together. So it makes sense if there's an injury that you would want to sort of get that structural stability in there as quickly as possible. So.
All right. So astrocytes, then we have microglia, or you want to go oligodendrocytes?
You said oligos first.
So oligodendrocytes are basically these hyper-communicative cells. They touch a lot of different cells in the brain, kind of similar to astrocytes. Again, they're very, very branched. They're very tree-like. And over here what you can see is you can basically see in red and green the cell body of one of these oligodendrocytes with... Did we decide that the green was mRNA or protein?
I think the cell is in red, and all the greens are like bodies of other folks. But that was just from another picture I found on Wikipedia. And
you can see all of these nuclei that basically the oligodendrocyte, they're these black spots because DNA isn't stained here, so you can't actually see the site where the DNA is stored. And so all that you can see is you can see these hollows where the nuclei of the other cells that the oligodendrocyte is wrapped around are located. And these are, in addition to the myelinating of the axons that they do, they're also kind of their recruitment cells, for example, for complement. They produce complement activation cascades, which are necessary for kickstarting the entire innate immune response.
And clotting.
Which will be important later. They produce cytokines and chemokines, and so when you have immune privilege, you have a really, really strong blood-brain barrier. Which
What's an immune privilege?
Immune privilege is that the brain doesn't get surveilled by the immune system, right? Mm-hmm. And so the reason that they figured this out is because they were like, look, if we take a cancer cell and we put it into a mouse body anywhere else in the body, the immune system appears to see it, it reacts to it, and it destroys it. But if we do the same thing and we put it inside of a brain, it doesn't seem capable of seeing that there's something there that shouldn't be there. And so they were basically like, well, the immune system must not be surveilled by, or the immune system must not be surveilling the brain. And it hasn't been until recently that they're like, well, caveat. So with the oligodendrocytes, they are part of the process by which this immune privilege starts to break down a little bit, because they're producing the kinds of cytokines that are inflammatory, that break the tight junctions. And these tight junctions, once they're broken, will allow other cells to come in. Like you get T cells, you get B cells, you get the lymphocytes like the neutrophils and the mast cells that are all able to kind of infiltrate the brain. And this means that these are massive players in terms of being able to create an absolute disaster, because you don't want inflammation in the brain. It can lead, like I think that it was Lily who mentioned encephalitis, it can lead to encephalitis, but more realistically, it can just lead to the destruction of really delicate cells that have a hard time regrowing. And so you really, really don't want to have a lot of inflammation.
Yep. Yeah, the brain, regrowing the brain is central to all of this, in that it's very difficult.
And then microglia.
Microglia are kind of cool. It looks like they are the closest analog we have to immune cells in the brain, like straight up immune cells, in that they seem to be more mobile than these other astrocytes that are very structural and embedded.
Well, they have a close parallel to macrophages, right? They're phagocytic cells. And so what this means is that while astrocytes and oligodendrocytes are immune cells in that they participate in the production of cytokines, they participate in antigen presentation, they communicate with the T cells, they activate inflammation. They're sort of these kind of innate cells in some way, because they're recognizing PAMPs and DAMPs, which are damage-associated molecular proteins, and damage-associated molecular patterns and pathogen-associated molecular patterns. They're also reacting to innate things like reactive oxygen species, to, I think, changes in like dopamine I saw somewhere. And so they're basically very, very much innate cells, but distinct to the brain compartment. On the other hand, microglia are basically neuronal cells that are macrophages.
Yeah, with some specialized roles and so forth.
And the specialized roles are?
Just repair. Like this one thing they talk about is the synaptic stripping. Like if you have a traumatic brain injury, you need to get rid of the neurons that are connected to stuff but shouldn't be anymore. Maybe you have an axon connected but no cell body to receive the message or whatever it may be. And so they have a few specialized roles, but essentially they're doing repair and signaling, and they can jump in there when there's an injury. But yeah, the damage-associated proteins are numerous. Basically, you're not supposed to have trash floating around in your body, like cellular trash, and if you are, it's going to set off alarms, whether it's in the brain or anywhere else.
Mm-hmm.
And these are like common patterns that your body's looking for: pieces of DNA, pieces of internal proteins that are turned inside out, stuff like that.
Then there's one more part of the brain's immune system that is not a glial cell. It's actually just straight up an innate immune cell. It's a mast cell. So does anybody know what mast cells are known for in the body in terms of what reaction? I see some head shakes. Do we have some?
Got any hay fever people out there?
Yeah, does anybody have allergies? Who's got allergies? So mast cells are basically what carries the histamine that causes the allergic reaction. All of these granules, this is an electron micrograph of a mast cell, which I pulled because what you can see here is that you have this granulated nucleus where all the genetic material is, and then the cytoplasm of the mast cell looks really weird. It's filled with all of these compartments, and these compartments are granules, and the granules contain immune excitatory molecules, everything from cytokines to histamine. And so basically, the surface of these cells is studded with immunoglobulin E, which is produced by B plasma cells. So these plasma cells produce vast quantities of IgE, which are soluble in the blood. And these are antigen-specific IgEs, right? So the B cell is producing them because it has encountered some kind of antigen that causes an allergic response. It goes through the sort of the selection process to make sure that they're really good antibodies. It undergoes class switching. It starts to produce IgE. And then the mast cells, the surface of the mast cells is covered in something that's called the Fc receptor. And the Fc receptor is capable of binding the constant region of IgE. And so what happens is that the mast cell is then floating around the body, basically covered in IgE. And as soon as it encounters that same antigen that activated the B cell back in the day, which could be ragweed pollen, it could be dust, it could be cat hair, it could be whatever, then it activates this cascade downstream that indicates it is time for the cell to die, to lyse open, and to release all of these granules, which is why you have a histaminergic response. And when you take antihistamine like Benadryl, what happens is that you don't have the response because it tamps down your mast cells.
Yeah, another molecule that's on there that's kind of interesting in addition to histamines is heparin.
Which is a blood thinner. And heparin is a blood thinner, right?
Yeah, it's an anticoagulant. So it's going to stop some of those clotting processes. So you can imagine at another point for things to get dysregulated, especially when it comes to COVID, which we're not really touching on yet. But obviously, if these mast cells are dysregulated, you could have something contributing to the coagulative thrombic disorders.
And I mean, you'd want, in the case, if you are a mast cell and you're like, hey, I'm going to react to this antigen that's here that I've discovered, you want to release not just histamine, which is the it's basically a chemokine in the sense that immune cells are attracted towards it. You also want to release something that's going to prevent the clotting cascade from blocking the holes that you're making in the vascular system as passage into the tissue. And so heparin is there in order to make sure that at least locally you're not going to get any kind of platelet cascade that is preventing successful penetration of the immune cells to the site of the antigen entry into the body.
It's also important in creating new blood vessels too. So if there's a site of injury, you have heparin interacting with other factors, proteases, other factors that are allowing for the new blood vessels to move in and be put in place.
Mm-hmm. This is kind of a really rough schematic that basically is showing the way that the mast cell is. So this is exactly, it's showing that mast cells are involved in neurogenesis because it's basically activating cellular pathways that induce the growth of neurons. But it's also they're also involved in neurodegeneration and the increase in blood-brain permeability, because as they produce histamine and tryptase and this chemokine L5, they're causing the microglia to then create their own set of cytokines. And that set of cytokines is then acting back on the mast cell. And the mast cell is releasing all of these other things that are affecting its environment. And in affecting its environment, the neurodegeneration happens on the basis of basically activating this glutamine transporter, which then activates astroglia to produce another aggravating inflammatory factor, which is nitrous oxide, and nitrous oxide causes cell death in the microglia. And so it's basically this sort of like closed pathway where you can see how you can very, very easily get into a bad loop if your astroglia are suddenly losing the ability to, instead of sending an inflammatory signal, to start sending a de-inflammatory signal. You can see you can quickly end up in a position where things spin out of control.
I just want to add, we've probably said it before, but the blood-brain barrier is not like the Berlin Wall. It's not just in one place down like at the start of your brain.
It's actually in 4 places.
Nice. You made a slide for that.
I did.
Awesome. Okay, so this was kind of unclear to me when I was young, really young. I always imagined when I was a young chap. I always imagined the blood-brain barrier as this like thing that sat in between your brain.
Checkpoint Charlie.
And it is not. It is everywhere in the brain. Well, everywhere there's blood. So Go ahead.
All right, so basically the blood-brain barrier is technically in 4 places, but as an adult, you only have it in 3 places. So, and this picture on the left is on Wikipedia. When we put these slides together, we will pull a lot from papers, we'll pull a lot from Wikipedia. There's always a citation on the slide if you want to follow up or you want to do more reading or you think that we're moving too fast and you want to follow it up on your own.
We'll upload the slides after class.
Yeah, we upload the slides. You can look at them.
And we upload these lectures too. So if you all don't know about it, there is they're linked in the syllabus. There's a YouTube channel. You can watch these lectures again if you didn't get enough the first time.
Indeed. Okay, so we have the blood-brain barrier is always about tight junctions. So we talk about tight junctions in context of the ability for these immune cells to squeeze through cellular layers, to get out of the blood vessels, to get through the endothelial cells, and to get into the parenchyma of the organ. And normally these tight junctions are able to permit to stop the flow of water, let alone the flow of cells. And so when we talk about there being a blood-brain barrier, what it is that you have these layers of endothelium that are on the inside of either blood vessels or on the outside of the cerebrospinal fluid envelope that are so tightly bonded to one another that they don't let anything through. And so there's 3 of these that appear in adults, and that is, number 1 is the tight junctions of the endothelial cells of the cerebral vasculature. Cerebral vasculature is literally just the blood vessels of the brain. So when we talk about this over here on the left, you can see cerebral vasculature. This is a blood vessel. The endothelial cells are the ones that are the white boxes, and they don't let anything pass through them that isn't properly fitted to a receptor. There's no passive flow of anything across them. These are lined with 2 different kinds of cells, which are parasites, which are basically these kinds of barrier cells that line the vascular system. So when you have a normal condition, you have the what's the space inside a vascular cell called?
A space inside of a vascular cell?
Or like, what's the vascular space called?
The intraluminal space?
The intraluminal space, thank you. So the intraluminal space contains the blood, it contains the lymph mixed together. So right, this is very important to remember. Your blood and your lymph are mixed together inside of the body. And when you get to the brain, you're basically stripping the blood away from what amounts to the lymph fluid that's acellular. And that's what gets into the brain. It's the cerebrospinal fluid. So it's a fluid portion of the blood volume that is being pushed into the brain. And these parasites are basically the cells that are just on the outside of the vascular system. And so they help maintain the blood-brain barrier integrity by virtue of acting as this very tightly interlocked layer on the outside of all of the blood vessels. They're also involved in all kinds of different processes, like we talked about heparin being involved in angiogenesis when there's inflammation. These parasites are helping produce angiogenesis under normal conditions. They are phagocyting whatever's in the environment that shouldn't be there. They are helping regulate the cerebrospinal fluid. They're modulating the immune response, and they're also producing multipotent stem cells. So they're basically under controlled conditions. They're helping ensure that the blood-brain barrier is stable. In the same way, when things start to break down, that's when you start to see these neurotoxic molecules leaking through the walls of the vessels. You get problems with blood vessel formation. The phagocytosis isn't happening properly, and so you're accumulating a lot of trash at the barrier between the endothelium and these parasites. You get basically just gross dysfunction of the membrane, loss of immune privilege, and full-scale aberrations of the brain. And this is one of those places where when we talk about COVID, this could very well be one of the locations of dysfunction that we're seeing, because what we're seeing is brain dysfunction, not really viral dysfunction.
Yeah, I think if you look at this picture, it's kind of clear that the parasites sit on the blood vessel and squeeze it physically. I'm not saying that they're doing all the work of the contractile stuff, but they're certainly governing the blood flow. And there's increasing evidence, it seems, that parasites are important in hemostasis in capillaries in general.
Which ties them into the angiotensin system.
Exactly, and ties them into just leakiness and problems with, getting trash out of the brain, getting immune effectors to the right site, things like that. So...
astrocytes are the other types of cells that are part of this barrier, and you can see that basically you have these parasites that are right on the outside. The astrocytes lay on top of them on the outside of that, and they help maintain the blood-brain barrier integrity as well. But in addition, they're also basically innate and adaptive cells. They're, I guess you could relate them most closely to kind of like dendritic cells, right?
Who are we talking about?
Astrocytes.
Yeah.
Where they're basically interacting with all of these PAMPs and DAMPs. They
very much look like dendritic cells too.
Right? So they're immune surveillance cells. So they are stretched all throughout the brain. They have pattern recognition receptors on their surfaces, which means that they can respond to all of these innate issues. As part of their innate immunity, they're able to produce cytokines, and as part of their adaptive immunity, they're talking to T cells and they're helping mature them. And so, as far as I can tell, that's basically a dendritic cell.
Very similar, except it's in the brain.
And they're helping maintain the blood-brain barrier.
So some of those, yeah. It seems like the dendritic cells actually have the ability in the body too to migrate. I'm not sure that these do in the same way.
That
dendritic cells are often migrating to those secondary lymphoid organs.
That's true.
And doing some work there to get the B cell process going.
Mm-hmm. So the other place where we have another blood-brain barrier is what is called the choroid plexus epithelial cells. So again, these are epithelial cells, not endothelial cells. And here what we see is that we have a group of cells that are actually responsible for the production of the cerebrospinal fluid. So, and this is a really interesting thing because the blood vessels here are permeable and they release the fluids into the stromal space. And then these epithelial cells from the stromal cell space are the ones that are controlling what gets into the CSF. So they're said to produce the CSF, but what's really happening is that they're basically filtration cells. You are filtering blood into the stromal space, which I think I have a couple of pictures here. Yeah, so there's 4 different ventricles in
Let's just maybe like 30,000 foot view of what CSF is versus blood. So the brain has its own circulatory system, which is kind of fascinating, which I think is an immune structure to some extent, because the brain is able to isolate itself from whatever sort of junk might be in your blood this way. This is part of the idea. If you have really tight junctions and you have a blood-brain barrier, that seems like a great idea, except how do you do all of the things that blood does in your brain without having blood accessing all of those places? And so this is the solution that Mother Nature has come up with, which is that you're going to have your own kind of in-house blood-like stuff, which is called CSF. It isn't blood, but it's a fluid bath that's capable of transporting waste products and so on and so forth.
And honestly, as far as I can rationalize it, I think that the necessity for this comes down to the fact that you need to have a huge density of capillaries inside of the brain in order to basically produce the sort of nutrient and oxygen exchange that's absolutely necessary for metabolism to continue. But you also need to be able to make sure that the capillary, that there's this ability to prevent clotting from happening. There's this ability to prevent buildup of junk directly against the cells, because you want there to be a container where if something happens for at least a little while, it's contained within it before it spills out to the larger brain so that you have time to fix it. And I think that that's kind of the reason for something like this to evolve is to create a secondary control to make sure that if something is about to go wrong, you have time to surveil it before it gets to the most delicate membranes, the most delicate tissues. The cells, these epithelial cells of the choroid plexus are said to be the ones that generate the cerebrospinal fluid, because there's these fenestrated capillaries, which is that blood comes in, and there's no tight junctions between the walls of those capillaries, which allows the fluid to get into the stromal space. From the stromal space, it passes through this choroid plexus epithelium into the cerebrospinal fluid, and these areas are called ventricles. And so these ventricles are filled with the fluid, and the fluid washes all across the brain. So this is a continuous fluidic system of the brain. And you can find cool videos online of basically how in an fMRI machine, as you sleep, your blood flow goes down in the brain and the cerebrospinal fluid washes in, and then the blood washes in and the cerebrospinal fluid washes out. It's really cool.
So the CSF is produced right there too in the choroid plexus.
Yeah.
I don't know if you mentioned that, and it's also quite a bit every day. It's like a can of Coke's worth of CSF.
But when they say produced, like for me, I'm like, what does it mean produced? It's just filtered. It's like it's a filtration system. You have fenestrated capillaries that leak.
You ever see those little signs on the water fountains that say like, saved this many bottles of water today? I think it's the same idea. It's like it didn't really produce the water, but it did sort of give it out.
Yeah. I think that's a good way of looking at it. Oops. So that's pretty straightforward. This is obviously blood-brain barrier because you are preventing the stuff that's in the blood from getting all the way to the CSF. It can get stuck in this stromal space, which then gets emptied out in and of itself. And then finally, you have one more tight junction, which is between the endothelial cells of the arachnoid vessels. And the arachnoid vessels are basically these areas that are filled with extraaxial cerebrospinal fluid. And so you have a layer that's here also that is at a different part of the brain, and it is not at a blood cerebrospinal fluid interface. In this case, it's a cerebrospinal fluid interface, as far as I understand it.
Yes. Another really interesting role for CSF is that it floats the brain. So it reduces the weight of the brain by something pretty significant, like an order of magnitude, effectively. So it's like a cushion, too, which is pretty cool.
Who knew? The interface between the immune system and the brain, though, was not really discovered until relatively recently. So this is actually a paper from last year, February of last year, where they have pinpointed the location inside of the dural sinus, which is actually where you get the most amount of immune surveillance, and that's where the immune reaction of innate and even adaptive cells kicks off before moving throughout the entire brain. And so this is a schematic. I think that this is from Gray's Anatomy, actually, or something like that, which shows you that the dural sinus is this vessel that runs through the entire brain. It runs through the entire brain surface, and it is distinct from this subarachnoid space because the subarachnoid space stretches. It's basically this cushiony fluid that's everywhere, but inside of the head case, inside of the skull, up against the bone, there's basically a vessel. It's called the dural sinus, and it contains both blood and cerebrospinal fluid, and then as it leaves the body, so it basically is formed here in the center of the brain, circulates through the brain, and then it leaves the head by draining into the internal jugular veins, which is why I'm saying that the cerebrospinal fluid is produced by the choroid plexus in name only, because it's mixing with the blood, and that same blood circulates back up to the choroid plexus.
I mean, it's basically blood without the red blood cells.
Yeah, exactly.
It's missing some proteins and stuff too, but...
And so at this
It's
confluence
plasma.
of sinuses, basically at this crossroads, is where you have kind of like a lymphoid organ. You have in the sinus a bunch of immune surveillance cells. You have T cells there, you have B cells there, you have neutrophils and macrophages, and they're all hanging out there waiting to see if they find some kind of innate signal that something is wrong for them to start acting inside of the brain. Overall, this is from this same paper that came out in 2001, and it's a schematic of the way that this all works, which is the fact that you have some kind of antigen that is coming from inside of the brain, right? So you have a central nervous system antigen that is getting into this subarachnoid space, and then from the subarachnoid space, which is the cerebrospinal fluid, it crosses the dura, and then it gets into the superior sagittal sinus. And the sinus is basically this vessel that runs all the way through. Like if we go back to here, the superior sagittal sinus. And so we're looking here down the axis of this photo here. So it's like a cross section through the skull, like this. And once this antigen has reached the superior sinus actually has a pretty robust collection of immune cells that are there and available to do it. So you can see that we have these yellow cells that are T cells. And so there's antigen presentation to these T cells. Once the T cells have undergone antigen presentation, then they recruit...
With the dendritic cells.
With the dendritic cells, yeah, exactly. Then you have this leukocyte recruitment. After you have the leukocyte recruitment, then you have these hubs of immune activity where you have macrophages and T cells interacting, attempting to deal with the antigen that has arisen, and we are a go, right? From here on out, once you start breaking these tight junctions, if inflammation continues, that's when you start to have pretty severe consequences of everything moving closer and closer to the site of the brain that's actually inflamed. We kind of talked about the flow of this comes from the choroid plexus through the ventricles. I just thought that this was an interesting diagram that kind of shows the intricacy of it as it moves through the brain. But all of it flows out, drains into the jugular veins, and then is recirculated through the blood vessels back through the center part. And this is, I really wanted to show you guys this. So this is called, oh no. This is called lymphatic flow. And I couldn't embed it because it was an OGV file, and I don't know what those are. But basically, I'm going to pause this really quick. This is the glial lymphatic pathway. When we talk about the fact that the cerebrospinal fluid washes the brain and it clears things out of the brain, we are literally talking that there is a flow of fluid through the parenchyma of the brain that runs across the cells that are there and takes all of the metabolic wastes and brings them to the drainage sites and then pulls them out. And so this is basically a little video that runs. And so you can see that you have this wave of lymphatic fluid that runs across the cells in the brain and carries away all of the metabolic wastes, which are represented by these little black dots.
And glial is just a general term for all of these cells that we've talked about at the beginning, for the most part, like the astrocytes, microglia. They are myelinating cells. They're structural cells in the brain.
Exactly. And so this is just to sort of emphasize the way in which all of these pieces come together in order to create a functional brain space that is both cleaned out, maintained, and protected from inflammation. The neurological effects of COVID, in my humble opinion, have a lot to do with the interruption of the lymphatic flow. Like, we'll talk about this beta-amyloid paper in a second, and we'll talk about the neurological effects of COVID. But I think that we should take a break.
So I was gonna say.
Okay. Let's take a...
Then we'll do COVID, and as it relates to all of this.
10?
Sure. Yeah.
Okay. So we'll see you back at 46.
All right. So the first paper we're gonna check out is the nervous system consequences of COVID, which is a more sweeping overview of what's happening. And I want to just highlight some of the salient features as I was reading this. And then afterwards, we can start to discuss any thoughts or ideas you guys had as you read this too. So, right out of the gates, they're basically talking about these myriad neurological complications that are showing up: confusion, stroke, neuromuscular disorders are showing up during acute COVID-19. There's also all of these peripheral maladies that they list: headache, sensory disturbances, depression, even psychosis may persist for months after infection, which is, I think, why this is such a scary topic, because what could be worse than having your mind affected? I mean, obviously, having a lung infection is really scary, but not being able to think or react or behave properly is its own level of a nightmare. And they tell us right out of the gate that they don't totally understand the pathophysiology of how this happens, so we have to brace ourselves and recognize this is quite a new subject. We're not going to get totally to the bottom of it. However, they point out that nonspecific neuroinflammation and anti-neuroautoimmune dysregulation is likely at fault, and we're going to talk about some of the pieces of that as we go ahead.
And non-specific neuroinflammation in this case is basically everything that we've been talking about. It is this glia, the astroglia, the microglia, the oligodendrocytes, the mast cells, the T cells in the dural sinus, the macrophages in the dural sinus, the dendritic cells there. Everybody's just kind of on and unhappy.
Yep. And so they talk about everything from strokes to, like, microstrokes could be behind this, to acute inflammation, demyelination of central peripheral nervous system. All of these things are being reported associated with COVID. And this gets a little hairy too, because we run into the weeds with cause and effect. Obviously, you're looking at a huge portion of the population, closer than you ever have before. It's difficult to separate that out, and they actually get into that more in the next paper. So it doesn't only induce respiratory systems. We see multiple organ systems involved, which we knew of from before: kidneys, GI, heart, brain. And it seems like the most common neurological symptoms are anosmia, which is the loss of smell, stroke, delirium, brain inflammation, general encephalopathy, primary psychiatric symptoms, and peripheral nerve syndromes. And that these cerebrovascular problems can happen before the onset of even traditional respiratory patterns that we associate with COVID, the respiratory distress. This can happen beforehand, and they can also happen later. It says actually generally they happen later, after a couple of weeks. This is kind of interesting too here. Originally, we thought that there was speculation that the virus might be making its way into the brain, and that's essentially failed to manifest. And so some of the earlier speculation involved actual viral lytic processes in the cerebral tissue. Now we're looking for something else, right? We're looking for these secondary immune dysregulations because they can't find viral RNA in the CSF of these patients.
And this is also, it ties back to the idea of what Ava was saying at the beginning of class, which is like, look, I know somebody that has symptoms, but he doesn't test positive for several days. And it's the question when you see a report like this, which is like, well, we've looked and we don't see COVID, but we see symptoms. Is that because you haven't looked in the right place? Is it because you haven't looked at the right time? Is it because you're not sampling in the correct way? It just becomes very hard to evaluate. Is it because you're not looking for spike protein when it's the spike protein that's gotten in and it's not actually a PCR thing that it's not a nucleic acid problem that's in there? Like it becomes very complicated.
It also is complicated by the fact that in order to look at someone's brain tissue, they basically need to be dead. And so they note this in the paper that when we're talking about all these things that could go wrong in the brain, we're looking at people who had extremely severe disease or were killed by something else, which probably complicated the data. They say those who died with acute COVID-19 had severe disease that may not be representative of the majority of those infected with SARS-CoV-2. Many had systemic or metabolic derangements prior to death that may contribute to the pathology in a nonspecific way. However, when infrequently detected, infected cells in human brain lack surrounding clusters of inflammatory cells, suggesting that SARS-CoV-2 presence in the CNS does not incite classical viral encephalitis. So, again, it's not the virus directly.
So they're saying that even if you do find virus in the brain, which is vanishingly small because it doesn't appear to translocate in the brain, there's no cluster of immune reactivity around it. And
these tissues are really messed up when they start this examination. When they look at the CSF, though, from patients that are still alive, they see markers of neuroinflammation and markers of T-cell exhaustion.
Is that what we're doing next week?
No, next week we're going to do the endothelial stuff, or the vasculature stuff, and check out the renin-angiotensin system, stuff like that. But maybe we'll do T-cells the next week. Yeah, that's the whole. It's okay. That's a whole other line of thought. We haven't figured it out yet. More on that later. I mean, I guess this is worth an aside in case we don't get back to it. So, immune exhaustion is a huge piece of the puzzle in general, not just in neuroinflammation. But this is basically the idea that the immune system is juggling so many plates that it can't really be effective on any front in particular. And we've seen this. When we taught this class previously, we did some papers specifically on B-cell exhaustion and T-cell exhaustion. And it does seem to be a consistent problem in patients that have severe disease, that their T-cells aren't doing what they're supposed to be doing, and the B-cells aren't doing what they're supposed to be doing. And it seems to have to do with an overburdened state with regard to inflammation. So,
and I wanted to go back up a little bit. So if you go up above this pull quote, they say here that additionally, CSF-specific clonal expansion of T cells and antibodies that recognize epitopes of SARS-CoV-2 spike protein that cross-react with neural antigens suggests compartmentalization of the immune response, although the possibility of a persistent infection with restricted viral replication cannot be entirely excluded. And what they're basically saying here is they're acknowledging the fact that there might be active viral replication that is causing these problems, but it is restricted compartmentally. And so we talked about this a little bit, where it's possible for some viruses to evade the lysozyme. They get into a phagocytic cell, and then instead of dying, they're able to perforate the membrane and continue sort of doing the thing that they do inside of there, because they effectively decapsulate and then can start the process themselves.
Yeah, I'm glad you brought that up. I missed that paragraph up top. Thanks for pointing that out. They put this huge quote in the middle of this paper, and it makes it rather difficult to see where the page breaks are. Okay, so again, they're back to looking at autopsy data next. They see infiltration of macrophages, CD8 positive T lymphocytes. So those are the cytotoxics in the perivascular regions, which is what Dr. Bendebre was kind of walking through in that one of the last figures previously. Widespread microglial activation throughout the brain. So inflammatory markers being sent out, alarms. And again, in these autopsy patients, they're not seeing the SARS-CoV-2 RNA. But that doesn't mean it's not there. It just means they're not seeing it. Right. It also doesn't mean that it's not past or prior to the onset of the replicative phase for the virus that you're looking at this, because a lot of damage can happen from the inflammation alone before you actually have replication, which we talked about earlier. So the next question they want to ask is, what does the vascular deterioration that we've seen in other aspects of COVID, how does that contribute to the neurological paradigm here? And they note that acute COVID-19 is associated with heightened risk of stroke compared to, say, the flu. And so that's a reasonable thing to go and look for in the brain. Obviously, strokes in the brain, this is essentially a ruptured blood-brain barrier, essentially. Or you have some sort of blood clot from some other part of the body that's going and traveling to the brain, blocking off blood flow. And so, let's see, what did they find? They found increases of markers of vascular inflammation. They found markers of thrombosis and infarction. And so it seems like that is reasonable. Markers suggesting endothelial inflammation and coagulopathy, that's clotting, contribute to the pathology. And they are going so far as to say that it is plausible that micro blood clots in the brain are appearing. These are like sort of subdetectable levels just noted from various biomarkers, molecular biomarkers associated with them. Could be something, they could essentially approximate some of the less severe versions, some of the less severe symptoms. They could approximate microstrokes, essentially. So some of this stuff, the brain fog and all of that, could actually come down to these little ruptures, little blood clots in the brain. And this plays in a bit to the next paper we're going to look at with the amyloids, because amyloids are essentially little pieces of scarring that happen in the brain when it's injured, including when there's blood clots.
Which should be cleared out if there's proper circulation of the cerebrospinal fluid. And that's something really to consider, which is that the brain has the ability to clear this stuff out. And what it seems to be is that COVID is creating conditions where the system of clearance is getting disrupted.
It may be, yeah.
But as the paper says, there's severe disease already, so maybe it's already on the road to being disrupted, and you just add this extra stress, and the entire sort of house starts to fall apart from the foundations.
Right. So this entire cluster of symptoms that they're calling long COVID.
Yeah, it says here, like, many people who experience neurologic symptoms that linger after acute COVID-19 are less than 50 years old and were healthy and active prior to infection. Notably, the majority were never hospitalized during their acute COVID-19 illness, reflecting mild initial disease. And this is where it gets to be really hard to tease apart what's happening, because you can think your way into a massively inflamed state. If you are constantly under stress, if you are constantly...
You can think your way out of it too, right?
Yeah, you can think your way out of it. You can think your way into it. And again, I don't want to intimate that people are not experiencing something very, very, very real. But it's interesting that the people who experience the worst conditions are people who are young didn't have a particularly strong COVID response, and it's almost as if the aftermath is what is creating the mental fog. And I have a feeling that there is a non-zero contribution of the fact that people who are getting COVID at this point are people who have done everything right for many years. Do you know what I mean? They're people who have stopped going out with friends. They've worn masks everywhere. They have had all of the boosters. They have followed all of the rules. They've stopped touching their face. They've given up tremendous amounts of their lives for this. And to catch COVID still, despite all of that, I think is, at least from where I'm sitting, I think that it is at least a small contributing factor to the sort of stress that the body is undergoing, and it could contribute to the extension of an inflammatory phase because, I mean, I think that that's just straight heartbreak. Like, I think people can die of heartbreak. I really do believe that there are bodily states that happen. Like, you see somebody who's been married for 80 years. Not true. You see somebody who's been married for 50 years and their spouse dies, and then a week later they die, but they were healthy beforehand. It's possible to get massively sick.
And it's what's interesting about this long COVID too is it does seem to approximate a lot of other disorders that are these sort of vague, like fatigue disorders, right? That are now they're finding are the results of earlier injuries, right?
Mm-hmm. And so it's a combination
like Lyme
of two
disease
things,
they
I
mentioned.
think, yeah. And you start to get into these really, really pernicious places because medicine has unquestionably a tendency to dismiss people's concerns. Like, I read this really depressing article the other day about this lady who was like, I had ovarian pain for many years and I would always go to doctors, and the doctors are like, oh, you must just be stressed. Stress is really bad for you. And after, I think, 10 years of going to doctors and just being told that she's stressed, they pulled out like a 10-pound ovarian tumor from her. And so I think that there is this tendency to point to things and be like, you're stressed, you just need to fix yourself because there's this mind-body connection. But in reality, the mind-body connection is some small part of it, but there's also cases where people do get really tremendously injured, and it's incredibly difficult to figure out which is which because it's just such a delicate subject because you don't want to devalue anyone's experience. You don't want to point to somebody and to be like, oh, well, is there maybe some other reason that you're experiencing this? And then medicine, on the other hand, is perfectly happy to be like, oh, well, it's stress. If you just worked on it yourself, you'd fix it. And that kind of takes it off of their backs because, well, if it's just stress, then it's not our responsibility to fix you. You have to fix yourself. And so it's not a clear back and forth at all, I would say.
No, but that being said, it's probably not gonna hurt you to exercise and, keep your stress at bay. Breathing, social networks. So so they have a nice figure here. Can we show this figure maybe?
Mm-hmm.
This figure is probably not showing anything we haven't already talked about already, but it's worth mentioning. This is a really interesting paper on the level of we don't know exactly what's happening yet.
Which is why
it's actually much less of a nightmare than the first time we taught this course, which was in the fall of 2020, I believe.
At the fall of 2020, we basically read molecular papers every single time. And
it was the first papers that had ever come out about any of this, and a lot of those papers terminated in because it was so early in the process. And obviously this unraveling this long COVID business we're going to see a lot of papers that terminate in, well, it could be this, it could be that, but we're not totally sure yet. And I think that's what we're looking at. You got to understand some of these chronic diseases like chronic fatigue syndrome, Lyme disease, they're still heavily debated in terms of what's causing them. Even as we start to think we might be closing in on some answers, there's rational arguments from multiple perspectives in all of these disorders. They're very difficult to detect because it's likely that they result from an injury that occurred in the distant past, and you have a cascade of pathologies that result from that.
And there's this one little piece of this. So overall, this figure is basically reiterating the fact that mechanisms leading to injury are unknown. There's not really spike protein in the brain, so we don't know how that's been happening. There's all of these different ways in which we think that blood vessels might be damaged, but we're not 100% sure. We definitely know there's coagulation. And then, there's this generalized neuroinflammation.
Which we're going to do a ton on next week, by the way. I realize that that's something that I want everyone to leave this class understanding is what is going on with blood vessels and COVID. And I think it's worth a whole week. So I just uploaded the paper for that, by the way. So it's a pretty deep dive, and it will feed into what's going on in this figure too, because we need to understand what that actual epitope, the ACE2 system is supposed to be doing in the body, and how all of these different effects, both from the virus and from the therapies and from everything, how are they playing into this system? And so understanding this figure is going to require us understanding the native infrastructure for that renin angiotensin system, as well as how the virus is modulating it.
My sort of takeaway from here, though, is undetermined host factors for susceptibility, which is a single throwaway sentence in a big paper, but I think that it is super important because I really enjoy scientific theories that put possibilities down on the table. And right now we have a version of viruses, which is that the virus is dangerous to you, and there's a percentage by which you can evaluate how dangerous it is based off of like what group of people that you're binned into, whether it's by age or by bodily condition. But there's this other aspect of it, which is the fact that your environment and your relationship to what you are surrounded by is going to influence your ability to react to a virus. You can imagine, so there's a theory that's floated around for a long time in the aftermath of the big pandemic during the early 1900s, which is that, yes, the Spanish flu, unquestionably dangerous. The guy whose name I don't remember, I think was like Tarek or something, he just recently died. For a long time, there was this open question. Why was the 1918 flu so dangerous? Why did so many people die? And this guy figured out that there was this colony in a town called Breivik, Alaska. He went out there. He got permission from the I want to say that it was the Inuit who lived there to exhume this mass grave that had been dug into the permafrost in the aftermath of the 1918 pandemic because they lost a huge number of people. And he dug out these bodies with their permission, and he got a biopsy of somebody who died of the flu. He got a biopsy of the lung. He brought the biopsy back home, and he managed to actually cultivate the original virus from 1918 from this frozen tissue. And when he was able to do that, in the lab, they were able to look to see what it did when it was introduced into cultured cells, and they saw that, in fact, it did activate a cytokine storm. It was a more aggressive version of the flu than a normal seasonal flu. Okay. However, you're still left with the question of, yes, but why did so many people die despite the fact that it was worse? Because we're talking, how many million?
Well, you got to understand, too, there's this mess that was happening called World War I.
Well, that's where I'm leading to. Hold on. So, I'm going to look to see. Spanish flu.
By the way, was this was recently he dug up this thing?
Oh, this was in the 90s.
I see.
Okay. So, they think that 500 million people got sick, and they think that 50 million people died of the Spanish flu. And what Dr. DeLay was about to say was the fact that, look, the Spanish flu immediately came after World War I, when people were incredibly malnourished. They were exposed to the horrors of...
The supply chains were wrecked.
The worst war of memory. There was massive famine. There was no way to get food. There was no way to get clean water. And so people were already really sick. And so I wonder if what we're looking at here when it says undetermined host factors is not the fact that our environment right now is filled with a lot of pretty unhealthy stuff. Everything from, you go to the grocery store and if you look at the ingredients list, there's artificial flavors, there's artificial colors, there's all kinds of stuff that is in the food supply that is coming from questionable locations. There are all kinds of chemicals in the water, like, if you walk down the road, you can see that there's lines of tire tar in the asphalt. And every time it rains, that stuff washes off into the drainage basins. In the drainage basins, it gets into the environment, eventually gets into our own water. And so the question is the severity of COVID-19, at least in part because there are people who are so sensitive to the environmental degradation that we currently find ourselves in, that they are having these massive responses to a virus that is otherwise not that dangerous. And we all know that there are people that we encounter that are more sensitive than other people, and there's no reason why this doesn't extend medically. The same person has one level of tolerance for pain, another person has a different level of tolerance for pain, and it's the same stimulus, but the outcome is different. And so everyone is exposed to the same environmental conditions, but it's entirely possible that what we're seeing in terms of severity of COVID is what people are most susceptible to the environmental conditions that they find themselves in. And that is really the deciding factor that is pushing people towards these incredibly severe syndromes. And so it's through no fault of their own. It's through nothing that they have done themselves. It's just that they have a constitution that is more sensitive to the environment in which they grow. And medicine doesn't yet have a way of dealing with that because it's too big of a question. How do you create a control group for someone that's not exposed to the kinds of environmental degradation that we're all surrounded by, who's not being showered with microplastics in the rain and there's no, prescription drugs in the groundwater? That control group doesn't exist. And when you come up against the point that you are looking at a situation where you know that it is variable based off of interaction with the environment, and you know that the environment is not particularly healthy, that last little bit, that last line of detail is going to be incredibly hard to figure out because we first have to find out how to ask the question. And so as you go forward in your lives and you move into medicine and you move into graduate programs and you move into jobs and you carry with yourselves this understanding of immunology and the environment and biology, see if you can find ways to tease that out. How do you understand the ways in which the water in which we live and the water in which we swim, how do we come to understand the effect that that has on us in order to be able to affect lasting positive change in an evidence-based way?
Yeah, right on. So I think, if I understand what you're saying, it's there's a lot more to this than just the virus itself, and that's really interesting. And I think it doesn't get the airtime it deserves. Viruses are like we said in the first lecture, they're all over the place all the time. They're not usually killing us. They're not even usually making us sick. And
I just can never get over the fact that even with Ebola, that has a 90% case fatality rate, there's some 40% of people that just never even develop a case of Ebola.
Yeah, they can just hang out with Ebola people all day long and they're fine.
And that is the greatest mystery, because once we come to understand those differences, then we can really start to be able to say something about personalized medicine. That way we can really start to develop risk profiles for individuals so that they know exactly what it is that they should be worried about versus things that they don't have to be worried about. And that is kind of the future.
It's a basic biological question too. It's a lot of our investigations are motivated by medicine, by trying to heal people that are sick, not necessarily try to figure out what's going on in nature. You guys will figure this out soon enough if you go into the research. It's very difficult to just study something because you think it's cool or interesting. You have to very much motivate it with something instrumental in order to achieve funding to do that study. And so viruses are always going to be studied from the angle of their pathology rather than what other services they might be providing in nature. And
I think that
this will take a while to untangle.
I think that is, I would go so far as to say that it's a fatally flawed approach to biology.
Gloves are off. Yeah, I agree. Well, it played out. It's worth mentioning again. We mentioned it, but we saw this play out with the microbiome previously, right? Where bacteria were bad for a long time. Now bacteria, it's like, well, there are bacteria that do stuff. We need them actually, And I think that'll be a similar level of understanding that has to emerge.
I have this feeling that when huge amounts of resources are put into solving a question, like they have been towards COVID, right? Like there's no question that over the course of the last few years, there has been more money put into COVID than basically into any other kind of biology. Maybe cancer research has still been getting the same kind of attention, but huge amounts of money. Like everybody who is able to pivot to studying COVID in some form or another has done it because it's easy enough to get grants. And despite all of this, we haven't come up with a consistent model for why it's so bad. And to me, that would indicate, after all of this time and all of this attention, that we're not asking the right questions. And so as you guys do your research project and you start thinking about these questions and you start going into this, start really trying to focus on this larger goal of, well, in order to get to the bottom of this, what kind of questions would we have to ask? What would it look like to understand if the invisible things, the things that we cannot see, that we do not know, if they are affecting us? And how would we tease that apart? I think I would really recommend that.
I think that we should just briefly mention the other paper.
Okay.
Can we jump to those slides maybe?
Yeah, let's do it.
Yeah, so the other paper, it's interesting. So this is a group that thought, okay, we check out all of these autopsies of these severe patients that died with COVID, severe COVID patients that died. We look and we actually find that they have these amyloid plaques in 10 out of 10 of these autopsies from these severely ill hospitalized patients. They're looking for actual damage that they could potentially address therapeutically. Like, hey, if long COVID is coming because of the accumulation of plaques, well, great, we can find some sort of anti-plaque treatment.
Right, and they basically, the way that they came together with the fact that they had these... Are you going into that right now? Sorry.
I don't know what you're going to say.
No, go ahead.
I think that I just want to, before we even talk about what they found, they're very cautious to say that, hey, look, these are extremely sick patients, and they've also had traumatic brain injuries in the past, which are known to cause plaques as well. And so take what we're finding with a grain of salt. I saw these headlines in the news too, and after reading this paper, I'm like sort of less like sure that this is a good target or not.
And I think that that's part of the reason that I wanted to showcase this paper, is because I think that it's very easy to encounter a news headline and to be like, oh, well, it's a news headline.
Like they're finding these plaques in severe COVID patients. Wow, that's pretty interesting. But then you go, and these are a picture of the plaques, by the way. They're essentially, they're exactly what they look like. They're proteinaceous little scarry things. The size just depends on the way in which you cut it, mostly. But when you go and you look at these plaques in the world, you find that that most people are going to get these plaques if they live to a ripe enough age, they're going to get these plaques.
What do these numbers and letters mean?
Okay, so these are patients that don't have dementia. Obviously, we know
Which
that
ones?
The blue line is people with normal cognition. Severe cognitive impairment is SCI, and mild cognitive impairment is in the yellow.
So mild cognitive impairment has a higher percentage of amyloid positivity than severe cognitive impairment?
Yeah, isn't that interesting? So without making a class about Alzheimer's, these are thought to be a contributing factor. But obviously, again, it's quite prevalent in the patient population, in the healthy population to begin with. So while 10 out of 10 people aren't developing them, it just gets to be a little tricky, especially the case study here. This person had a history of a mild traumatic brain injury previously. It looks like the patient also died of something like a drug overdose as well, if I'm interpreting what they're saying correctly. And so it's like, I don't know what's happening here. It's I'm just not like particularly convinced after reading this that it's necessarily COVID related.
I think that the authors themselves are not necessarily convinced, and they're just kind of like, hey, look, this is a thing. Maybe it actually is a thing that forms when you're hypoxic under any kind of conditions, and it's going to require a lot of brain autopsies in order to figure out if this is actually specific to COVID or not.
And autopsies are problematic, of course, because the patient's dead. They were severe and outliers to begin with. So they also make this nice little line at the end too, which is interesting, that some of my highlights got kind of messed up. They basically found, let me see if I can find this. They found a secondary, they found that they couldn't detect a secondary marker with these beta amyloids. And that's important because the secondary marker, I can't remember what it's called right now. It indicates that the amyloids have actually started to bundle together and create macromolecules. So they say, as tempting as it is to extrapolate our results to patients who recover from their COVID infection, i.e. long COVID patients, that is not necessarily justified. And I think that's because these primary plaques are easily cleared out as opposed to the bundled plaques that actually create these aggregates that you see, these fibrils, which is, I think, what we're seeing on the screen right now.
Right. So, I mean, there's basically a collection of proteins that begin to accumulate inside of the neurons. And inside of the axons, they start to produce these amyloid beta proteins in large quantities because it's basically a SNP complex. So there's these three proteins that when they come together, they can break proteins at exactly the right place in order to generate something that is then sticky in the brain. And it's sticky in the brain not because it is something special enzymatically. I literally think that it's just a snarly shape-based problem where you basically have little chunks of protein that are electrostatically favored to interact. And when they're inside of the cell, you can have a body of them that is relatively small. And then if that cell dies and you have your glymphatic system effectively washing through, it can take the little thing and it can carry it along. It can get it to the venous drainage site. And then you basically, maybe your sentinel sinus cells don't even notice that it's there and they don't even react to it, because it's just basically like protein waste and nobody's really monitoring it that closely. But then what happens is that at the point where you either have an interruption of your glymphatic drainage or you have sufficient numbers of these plaques that are being formed in a sufficient number of neurons that once they escape, they basically are aggregating into larger and larger clusters, then you can imagine it's the same thing. It's like if you have like a little knot in a string, it's easy enough to undo. But if you've ever taken on a knitting project and you've accidentally unspooled a tremendous amount of yarn and then you just have this thing, or like a hairball from a sink, it immediately becomes much more difficult to clear out once you have large quantities of it.
Yep. I found the name of that secondary marker is called thioflavin, by the way, which indicates the oligomerization of these monomers.
And the oligomerization is basically the production of the large clusters.
So the authors are sort of hopeful that patients who survive the disease, who don't have severe disease, will clear out their plaques in a natural way. So maybe not. Maybe this is contributing to the long COVID, maybe not. We'll need more data.
I
I think we should stop and take questions though, since we only got half hour left or something.
think so too.
All right.
All right, let's do it.